event today. And first of all, this idea of this webinar was decided in the first meeting of committee chairs with SAFOG leadership, and the picture is of that meeting. And this good clinical practice SAFOG Research Committee Initiative webinar is going to be held today. And I would like to welcome you all, the SAFOG leadership, the FIGO president, FIGO past president, and very most of the renowned people, and then the participants. And this is the program. I'll be talking about the introduction program and chairs. And the chair will be the president of SAFOG, Dr. Rohan Atatua, and he will be also welcoming us. And co-chair is the General Secretary SAFOG, Professor Rashid Latif Khan, and then followed by an overview of good clinical practice by Professor Genath Baral from Nepal. And then having clearly defined research questions and appropriate study design will be dealt with by Dr. Malitha Patabenge from Sri Lanka, and conforming to overall ethical principle will be taken by myself, and quality assurance monitoring of the clinical trials in GCP will be dealt with by Professor Dr. Saida Batul Mazar, and ethical consideration for including pregnant women in clinical trials will be dealt with by Dr. Indu Asanka Jayavardhani, and collaborative platforms for clinical trials will be dealt by Professor Dr. Mohan Kamath from India, and there will be a question and answer section with discussion, and words of the wisdom will be given by Professor Sabaratnam Arul Kumaran and Professor Malik Gunavardhani from Sri Lanka, and comment will be given by the chairpersons, Dr. Rohana Atatua, and then closing vote of thanks will be given by General Secretary Professor Dr. Yusuf Latif Khan. This is the overview of the program, and the general instruction about this webinar is all participants will be muted during the webinar. If you have any queries, please type in question and answer section, chat section. If you have any comments, please type there. Queries and questions will be addressed at the end of the webinar by moderator Dr. Indu Asanka and other resource persons, and we will be recording this webinar, and the recording will be available after the webinar. And email notification will be sent to the all participants, and this webinar is accredited by Nepal Medical Council CPD points. So these points will be provided to all the participants. I would like to welcome Dr. Rohana Atatua, the president of SAFOG and chair of today's webinar. He is consultant obstetrician and gynecologist, chairman of the 9 Wells Care Mother and Baby Hospital, Colombo, Sri Lanka, currently president of SAFOG and Secretary General of Asia Oceania Federation of OBGYN and chair of this webinar. Welcome, sir. Professor Dr. Yusuf Latif Khan is the Secretary General of SAFOG, and he is Professor of Obs-Gyne, Rashid Latif Hospital, Lahore, Pakistan, and he will be co-chairing today's webinar. Welcome, sir. And our special guest is Sir Sabaratnam Arul Kumaran, Professor Emeritus of Obs-Gyne, St. George University of London, Foundation Professor of OBGYN, University of Nicosia, Visiting Professor of Institute of Global Health Policy Innovation, Imperial College London, past President of FIGO, past President of British Medical Association, and past President of Royal College of Obstetricians and Gynecologists of UK. He was appointed Knight Bachelor by Her Majesty, the Queen of the UK, in her birthday honors list in June 2009 in recognition of his services to medicine and healthcare. So he is our special guest. Welcome, sir. Our next special guest is Professor Malik Gunavardhani from Sri Lanka. He is consultant of Department of Obstetrics and Gynecology, Sabaragamuwa University of Sri Lanka, Emeritus Professor of University of Ruhana, Sri Lanka, former professor in the International Medical University, Malaysia, former senior professor and chair of Gyne, University of Ruhana, Sri Lanka, former chairman, Board of Study in Obstetrics Gynecology, Postgraduate Institute of Medicine, Colombo, past president, Sri Lanka College of Obstetricians and Gynecologists, vice president, South Asian Federation of Obstetrics and Gynecology. Welcome, sir. Similarly, we have this clinical research and good practice committee of SAFOG. The committee is chaired by myself, and I'm working at Kathmandu Model Hospital, Nepal. And our co-chair is Dr. Indu Asankha Jayavardhane, senior lecturer in the Department of OB/GYN in Faculty of Medicine, Colombo, Sri Lanka. And we have 5 members from India, Pakistan, Sri Lanka, Nepal, and Bangladesh. Professor Dr. Meka Krishna Kumari is from India, Professor Saida Batul Mazhar from Pakistan, Professor Hemanta Dodampala is from Sri Lanka, Professor Gyannath Baral is from Nepal, Dr. Tanjim Sabina Chaudhary is from Bangladesh. So this is myself, and Dr. Indu Asankha Jayavardhane is co-chair of SAFOG Research Committee. He is board certified specialist obstetrician and gynecologist, senior lecturer in OB/GYN Medicine, Faculty of Medicine, University of Colombo, and honorary consultant obstetrician gynecologist, Dijoya Hospital for Women and National Hospital, Sri Lanka, consultant of Obs-Gyne, Navaloka Hospital, Nine Wells Hospital, Colombo, Sri Lanka. He is the moderator of today's webinar. Thank you, sir, for accepting the role of moderator. And Professor Dr. Gyannath Baral is Professor of OB/GYN, Kathmandu University, Nepal. He is currently the chairman of Nepal Health Research Council, which is a government body and the apex body of research in Nepal. He is also coordinator of Health Science Cluster, University Grant Commission, vice president of Goson Gyne Society of Nepal, executive member of Nepal Association of Medical Editors, and chief editor of our own Journal of Obstetrics and Gynecology. Professor Dr. Saida Batul Mazhar is chairperson OB/Gyne MCS Center, PIMS Islamabad, Pakistan, pro vice chancellor of Syed Zulfikar Ali Bhutto University, Islamabad, chairperson of Rawalpindi Islamabad Chapter Society of Obs/Gyne, Pakistan, member academic board of SOGP, past chief editor of Annals of PIMS and JSAFOMS, president-elect of South Asian Federation of Menopause Society, and she has a lot of publication in Index Journal, and her interests are in health education, maternal health, fertility, and menopause. Similarly, Professor Meka Krishna Kumari is a professor of Apollo IMSR, Hyderabad, India, senior consultant, Medicover Women and Child Hospital, Hyderabad, national corresponding editor, JOGI, and national coordinator of FOXI, vice president, Telangana Congress of OB/GYN, governing council member of ICOG, and president of Obstetric and Gyne Society of Hyderabad. Thank you all. Now I'd like to invite Dr. Indu to proceed further and invite our President SAFOG for welcome speech. Dr. Indu, please. Dr. Indu, we cannot hear you, sir. Please unmute yourself.
Correct. Sorry. Thank you.
Yes,
done. I think you can hear me now.
Yes,
yeah, there was a small problem with not being a host. Sorry, just fine. Welcome everybody to this webinar on Exclude clinical practice, and it's the start to. I hope it's going to be a start to a fabulous 2022. The next item in the agenda is taking of the chair and welcome by the president of SAFOG, who is none other than Dr. Rohana Hattotua from Sri Lanka. Sir needs no introduction here. Could you please take over the chair, sir?
Rohana sir, you are on mute, muted.
All right,
Please
sorry.
unmute.
Yes.
Yes, we can
Thank
hear
you.
you.
Yeah. Thank you, Ganesh and Indu, for the kind introduction. I will be brief in my talk. Firstly, I am happy to be.
It's 6 o'clock.
Sorry. Dr. Jane Conry, the president of FIGO, Professor Arul Kumaran, past president FIGO, RCOG and BMA, Professor Malik Gunavardhani, past vice president of SAFOG, and Dr. Ganesh, the chair of the research committee, Dr. Indu Asanka, co-chair of the research committee, executive board members of SAFOG, I see Professor Lubna Hassan joining in, and all the other members from SAFOG, and also I see Professor Shanta Kumari, the FIGO treasurer and the FOXI president, and all the other members from our member national societies, and speakers, moderators, and the chairperson. So thank you very much for everybody, and I welcome everybody for this webinar, which is the first webinar organized by the research committee of the SAFOG under the leadership of Professor Ganesh Dingal. I'm aware that Professor Dingal has been very busy preparing this webinar for the last so many weeks, making the program more attractive and beneficial to the clinicians, particularly the younger researchers in our region. And in this regard, Professor Dingal was very fortunate to receive the advice and guidance from Professor Sabaratna Marukumaran, who is the authority in research, and everyone looks up to him when it comes to any research project. So I am very thankful to Professor Marukumaran for participating today and giving the webinar much more value. I also appreciate very much the contribution made by Professor Malik Gunavardhani, past Vice President of SAFOG and co-chair of the Education Committee, who is an expert on research and is a committed teacher on research. His valuable input has enabled the organizers to select very valuable, excellent lecture topics for today's program. The theme selected is good clinical practice, and it is very appropriate and timely, as it has to be followed by every researcher and has to be very knowledgeable. Everyone should be very knowledgeable about it. So it is a good clinical practice, as you all know, is an international ethical and scientific quality standard for designing, conducting, recording, and reporting trials that involve the participation of human subjects, and provides assurance that the data and reported results are credible and accurate, and that the rights, safety, and well-being of the trial subjects are protected. Also sets the standard for the design, conduct, performance, monitoring, auditing, recording, analysis, and reporting of clinical trials. So today's program, Professor Ganesh has been able to get experts in the field of research from our region to deliver these lectures and make us more knowledgeable in this regard. I'm certain that today's program will be of immense value to our members involved in research and also for the postgraduates. And before I conclude, I would like, because I see first the FIGO president, Dr. Jean Conry, joining with us. So I don't think it is that, we should take the full value of joining. So I would like her to speak a few words and give your views on this program. If you are able to speak a few words for us, it will be of great, everyone will be very happy to listen to you. Thank you very much. And I'm sorry for this very short notice without prior thing. So thank you. Just a few words from you will be very greatly appreciated by everyone participating. Thank you very much.
Thank you, sir, for that that introduction.
Yes, Professor Jean Connery will speak a few words.
Oh, good. I'm unmuted. Thank you. What a fabulous program. And thank you so much for just inviting me and announcing this. I just wanted to say thank you for all the work that you're doing. And what you're doing is building on what I call the most important foundation of knowledge. And out of knowledge, we have what are the pillars of education. So educating everybody here, research, interpretation, and implementation, because the scholars here are going to really be able to interpret and implement that research for us. And then everybody here is part of advocacy and capacity building. So thank you so much for all the work that you always do and for really building on those pillars that we rely on. So congratulations to all. Thank you.
Thank you very much, Jean, for that very kind words and greatly appreciated. Thank you very much. Asanka, you can continue with the program.
Thank you. The next is a brief address by co-chair Dr. Yusuf Latif Khan. Sir, are you there?
Right. Yes.
Maybe
He has not joined us yet.
I think he has not joined yet, so we will move on.
Hi.
Hi, Yusuf sir.
Hi, sir. Hi, Dinesh.
Speaker. His next speaker in line for us is Dr. Malik Patabandige. He is an acting consultant obstetrician and gynecologist with Ministry of Health and has finished his MD with the PGIM here. He is a quite prolific young researcher in Sri Lanka. His research awards include Professor Sabaratna Marulkumaran Young Gynecologist Award with the first place in SLCOG at 2019, and he has published in many high index and peer-reviewed as well as index journals with a good H index of 0.8. Dr. Patabandige is going to speak to us about Dr. Patabandige is going to speak to us about having a clearly defined research question and the appropriate methods of study design, how it makes an impact on good clinical practice. Over to you, Dr. Patabandige.
Thank you, Dr. Asanka Jayawardena, and have a nice day to all of you. I think now I am from Australia. I'm having my overseas training here in Melbourne. Now it is almost midnight. I hope the other way around in South Asia and in the UK. So without wasting time, I will share my screen. Can you get the slides?
We can, Malita, we can. And if you go to full screen mode, please.
Okay, yes. Thank you. Yes. Is it in full screen mode now?
It is in full screen mode now. Thank you.
Yes. Basically, I am going to have a short talk on how to plan, how to formulate a research question and its objective, and to go for appropriate study design. This is for an early career researcher, how to enter to the field. How do we plan a research? That if we break down into simple steps, first we have to select the area. For an example, in obstetrics, postpartum hemorrhage, preeclampsia, or give me a minute, or something is happening. I will share it again. Okay. Okay, then we have to select the area. Then in particular in that particular area, we have to go for sub areas. For an example, if you go for preeclampsia, then there are risk factors, diagnostic tests, treatment, like that. We can break down, and each of these sub area we can further we can go for further sub areas, and then when we found a particular area of interest, we can do a proper literature search. The proper literature search will help you to understand what is already known on the subject and where is the uncertainty of evidence, and then to find out the research gap. This research gap is the first step to formulate our research question. Of course, a good study always starts with a well-defined or clearly defined research question. Research question is the state of the statement. That is the question of interest, or whatever statement that identifies the issues or the issue to be studied in a particular project. Of course, this research question has to be clearly defined, concise, and focused, and it has to be done well in advance to plan our objectives and to go for an appropriate study design. If it were the PICO approach, the well-known PICO approach, how to formulate a research question? If we go for PICO approach, that is basically for randomized controlled trials. There is an example of getting into research question, but as there are several study designs, not only the experimental, there are observational. With regards to observational studies, we can't apply PICO approach, but the basic things is still there to formulate a research question. The getting into a research question initially starts with general observation. The clinician researcher has a question, that means a initial question. Then he continues general observation, followed by repetitive or continuous proper literature search. Then he identifies the gap. Then he builds up a research question. There is an example that I have about that: starting aspirin at 12 weeks of gestation in women at risk of preeclampsia, whether it is useful or not. There is an initial question. Then somebody has done a proper literature search. Then he has found out that there is an according to the PICO format, there are participants, there is an intervention, aspirin or placebo, whatever, and there is a comparison group, and there are outcomes. So we can build up a research question according to PICO format, and it is going towards an interventional experimental studies or RCT. So I am going with going this lecture with 4 studies: RCT, cohort, case control, and then the cross-sectional. That is for your convenience. If I start research questions for each of these studies, I think this is Prof. Ganesh, can you share my lecture? Then I can.
Is it not shared from there?
Yes, I shared, but sometimes it is.
Started screen sharing.
Yes.
Yes, it is seen.
Yes. Is it in full screen mode? Okay. For example, if somebody is interested in studying reducing the incidence of postpartum hemorrhage, then the research question might be: does oxytocin reduce the blood loss following delivery compared with the misoprostol? Then somebody else is interested in preterm pre-labor rupture of membranes. So the research question for that particular area or research gap might be: does expectantly manage multiple pregnancies complicated with early PPROM or preterm pre-labor rupture of membranes have better outcomes? Then somebody is interested in ectopic pregnancy, then he has done a literature search and found out risk factors. There is a gap in risk factors. So he is going to just study to look for what are the risk factors for recurrent ectopic pregnancy, because there is a gap. Then somebody else was interested in postpartum hemorrhage, and then he has found out maternal women's experience. In women's experience, there is a gap. So he has formulated a question: do women who experience postpartum hemorrhage have a negative birth experience? Like that, the research question has to be clearly defined, addressing the gap. And this is basically the other side of our hypothesis. And we have to be practical. The PICO format or whatever the other questions for the non-randomized control studies is what we want, the clinician wants. Please do remember, what does she want or the patient wants. In your studies, try to include at least one specific objective to address her satisfaction, perception, experience, whatever. You may use validated tools or qualitative input, whatever. That is one additional point that I wanted to elaborate. Then from questions, research question to the objectives. The objectives has to be self-explanatory, clearly defined, and realistic. There are basically general and specific. General objective means there is a general aim to be accomplished through your research. Then the specific objectives that is related to the specific research questions to be answered through the project. There is a primary objective, which is usually coupled with our research question. There is a base fundamental goal, long-term goal, and there are secondary objectives that is to explore the other questions or issues, variables around the primary objective. Once you have conducted the research, you have analyzed, you have results, you have conclusions, and these results and conclusions at the end should match with your objectives. That is the aim. That is why we should have a clearly defined, realistic, self-explanatory objectives. We have to be very careful when we are formulating specific objectives. When we have a clearly defined research question and objectives, obviously the study design is already there. These are the examples of objectives, general objectives for the research questions I have mentioned earlier. So we are using these terms, what you call the action verbs, to determine, to compare, to assess, whatever, that depending on your research question. And this is just to summarize the types of study designs: experimental, observational. And again, this has come. I will do it for one last time. Last two slides. Yeah. And so the study design. So our first research question was the oxytocin, misoprostol, whether there is an impact on postpartum hemorrhage. Yes, that research question and objective needs a randomized control trial. All of these are published studies in British and American Journal of ONGs. And then the second one, there was first one there was an intervention, oxytocin or misoprostol, and comparison group, and it is a randomized control trial. Then the second one, there was an exposure, membrane rupture, and there is a comparison group with intact membrane. So, but there is no intervention. There is a cohort study. You can do retrospective or prospective depending on your feasibility. And then the third one, ectopic, recurrent ectopic, then risk factors. Of course, there are cases and controls. There is no intervention. There is no exposure. It was a case control, retrospective case control. And the fourth one, the last one, it is basically a survey, cross-sectional with a qualitative approach. They are going to, they were assessing the negative birth experience. You can have validated tools or qualitative approach, focus group, in-depth or whatever, but you cannot compare in between several qualitative studies, whatever. You can with the other studies, but you can just describe, you can just present your findings, which has a very important clinical significance when it comes to the practicability of our research in the quality of in improving the quality of maternal care, healthcare. So that is my short presentation. And for the research question, objective and study designs, if you have any questions, you can hit on me. Thank you.
Thank you, Malita, for that very comprehensive discussion about the research question. Dr. Baral, are you ready with the presentation now? Shall we go back to you?
Dr. Malitha need to off the screen share. Stop the screen share. Yeah, maybe Dr. Baral can now share his screen.
Thank you, Dr. Bar. I think you might want to remove your, you might be on mute. We can't hear you.
Yes. Please unmute yourself.
Okay. Yeah.
Okay.
Okay. Is it full screen now?
Yes.
Okay. Now, this was my second slide. I think now I'll be starting from here where I had left earlier. There are 18 members of ICH. 18 members of ICH, 3 founding regulatory members from EC, European Commission, and Japan and USA, and 3 founding industry EFA of the respective countries, and 2 standing regulatory bodies like Swiss Medic and Health Canada from Canada, 7 regulatory bodies from Brazil, Singapore, Republic of Korea, China, Saudi Arabia, Chinese Taipei, and Turkey, and 3 industries, Bio, Global Self-Care Federation, and IGBA. Now, the purpose of this ICH was to prevent unnecessary duplication of clinical trials and post-market clinical evaluations, and development, manufacturing, registration, and supervision of new medicines, and the reduction of unnecessary animal testing without compromising safety and effectiveness. And ICH observers are there, 2 standing observers like WHO and IFPMA, and 31 observers from different regulatory bodies, authorities, and they are altogether 715 experts spread in 34 different working groups so that they will prepare the guidelines, respective guidelines. There are 70 guidelines we call QSEM. Q stands for quality. There are 24 guidelines. S stands for safety, 15 guidelines. Efficacy guideline, that is 21 guidelines. GCP is one of the guidelines from efficacy. M stands for multidisciplinary, 7 guidelines are there. Electronic standards for transfer of regulatory information, common technical document, and standardized medical terminology. These are the guidelines made by ICH. And GCP is the one that is the clinical practice guideline. And there are 3 major success to come with GCP. Another common technical document that has to be prepared by all clinical trial investigators, and MEDRA, that is the medical dictionary for regulatory activities. That is highly specific standardized medical terminology, and it is used for registration, documentation, and safety monitoring. This is the CTD trial, common technical document, which includes all clinical and non-clinical reports in the brochure. And this is the efficacy guideline that I'm going to talk. This ICH GCP, that's E6, that is 6th guideline, 2nd revision, and it is the design, conduct, performance, monitoring, auditing, recording, analysis, reporting of the clinical trials, so that the data will be accurate and credible, and right and integrity will be maintained, and along with the confidentiality of the trial subjects. And it has there's a small booklet. It covers all within 8 chapters like glossary, principles, and IRBs, investigators, sponsors, clinical trial protocols, investigator brochures, and different essential documents like pre-start or pre-clinical, pre-trial guidelines. Essential documents are 20. During the clinical trial period, there are 25 documents, and post-trial, after termination or completion, there are 8 types of guidelines, so or essential documents. So altogether are 53. Wherever applicable, that has to be maintained by the clinical trial investigator and sponsor. And there are the 4 stages, 4 phases of the clinical trial. That starts from the preclinical one and safety for the phase 1 trial, safety and dosing, phase 3 safety and efficacy. So our efficacy trial is the GCP one, and then it goes to the post-marketing safety and efficacy evaluation, that is the phase 4 only, that is devised by the WHO. Now there are 13 core principles, like conduct trial according to the GCP, like protect patients and our subjects of the involved in the clinical trial. We have to weigh risk against the benefits, have adequate resources and information to justify trial, write a sound protocol, implement quality systems, receive IRB or ISC approval prior to starting the clinical trial, use qualified physicians and their supporting staff, and obtain informed consent or assent or from legal representative. There should be recording of the information accurately and appropriately, and protect confidentiality of the data and participants, and handle investigation of products appropriately. Investigative product means if we are doing the clinical drug trial, those drugs will be the investigational products. So the first one is the Declaration of Helsinki. That is about the ethical principle. That is the regulatory requirement that has been devised from the Declaration of Helsinki. I think this part will be dealt with or spoken by another speaker, so I'll skip it. Another is the we have to conduct trial only if there is benefit against the risk. Or there will be risk, obviously, some bigger risk will be there in every trial, but that this should be minimum and benefits should be more for the benefit of the people and society. And right and safety and well-being of the trial subjects should be maintained. And the core principle of GCP, another is non-clinical and clinical information should support the trial. That has to be clearly mentioned in the common technical document. And there should be compliance with the scientifically sound and detailed protocol. Without it, we cannot perform the good trial. And IRB or IEC approval should be taken prior to initiation, otherwise there will be breach with the regulatory compliance with the GCP. Medical care and decisions should be done by the qualified physician. That's why in GCP it is written that the investigator should be qualified by education, training, and experience to perform his or her task. So with like us, we are gynecologists and obstetrician, we are doing in our field. We cannot do trial, we cannot give decision in critical condition in the ophthalmological trial or dentistry trial. So everyone who is investigator, he or she should be qualified and qualified enough to give decision during the adverse effects and during the conduct of the trial. Now, informed consent should be given freely by the participants, and accurate reporting, interpretation, verification should be there, and all information should be recorded, handled, and stored in a way that allows its accurate reporting and interpretation and verification. And it's yet to protect confidentiality of the records as well. We have to conform to the GMPs and used for protocol. Like GCP, good clinical practice, they are good manufacturing practice as well for the investigational drug, and we have to maintain it's manufactured and handled and stored. And there should be quality control and assurance. Quality assurance should be there in principle, and quality control should be in practice. So we have to assure the quality of the trial. So to enforce this requirement of the GCP, we should protect the human subject. We should follow the informed consent that has 20 elements mentioned in GCP. Investigators' obligation is obviously there, and sponsors' obligation and monitors' obligations are there to have a good trial, sound trial, and ethical trial, and documentation, data handling, and statistics should be good, and record keeping and archival system should be there, and quality assurance should be there, and responsibility should be discharged by the IRB or IRC to confirm its good clinical trial. These are the requirements to establish, to enforce the GCP. And these are the things that will help us to establish GCP in our clinical trials, like IRC and IRB approval, informed consent form. We have to have the monitoring plan. Otherwise, our process may not be complete. There will be some deviations that has to be controlled by all the monitors and by sponsors, investigators. Safety plan should be there to report for the adverse effect and to control it. And there should be good decision-making power from the investigator, and proper documentation should be there for the accuracy and integrity of the data, and data management should be very valid and in all forms. And there is the shared responsibility of all regulatory authorities. Could be authorities would be international, national, or the local. And IRB and ISC should be there for the shared responsibility, clinical investigator as well, and all sponsor and contract research organizations and monitor. They all have to have the shared responsibility. But overall, the sponsor will hold the ultimate responsibility of clinical trial. In fact, if we do a clinical trial, we clinicians are the actor, like an actor in a movie, and director will direct everything to the actors. But our responsibility is there. But ultimate responsibility rests on the sponsor, or if it is delegated to the contract research organization, they will have partial responsibility. And main initiation, management, and financing is from the sponsor only. Now, sponsor responsibility are to maintain quality QA and QC, and educate investigator selection should be there. If there is no qualified or appropriate investigator selected by the sponsor, the trial will not be in a good way. So we have to confirm the IRC or approval is there or not, whether it has been well reviewed or not. The investigator product is supplied in the good way and in appropriate time, and whether there is a provision of safety of the information or not, whether there is adverse reporting system is according to the format, according to the standard guideline or not, and there should be all compliance for record keeping and auditing as well. So these are all responsibility from the sponsors. Responsibility from the IRB side, they have to safeguard all right and safety, and they have to see whether there is any amendment, any change, any deviation. They have to review it again. They have to ensure whether the qualification or education or experience of the investigator is quite good or not, or appropriate or not. They have to confirm their informed consent and all annexed documentation for the clinical trial, and they have to have very clear guideline on financial information as well. And investigator, all in all, investigators are there, and on the trial side, they have to have their own responsibility. And what are the, you can see it. These are the things that we have to report it from investigator sides. They have to maintain all protocol defined requirements. We have to ensure all trial subjects medical care. We have to ensure communications with IRB, IEC sponsors. The compliance should be there according to the protocol, and there should be investigation of products accountability should be ensured. And if it's a blinded study, there should be a system of unblinding as well in case there is some adverse reaction or adverse events. The record keeping and progress reports, safety reporting should be appropriate. And if there's premature termination because of some deviation or some adverse events, then it has to be maintained and communicated on time through the sponsor, and final report should be maintained according to the resource essential document. And contract research organization will help us, will help not for us, but it will help to the sponsors so that those all part of the sponsor responsibility will be delegated to the CRO, so it will be easier to conduct the trial in the trial site. You can see sponsors may transfer all or any part of the trial proceedings to the CRO, but ultimate responsibility rests on the sponsors only. Like GCP, there are other clinical practices, good practices, like if we do not have the good manufacturing practice, our investigation in a drug will not be in a good shape. If we do not have the good laboratory practice, our investigations will not be good. And if there is no good documentation practice, our clinical research will not be in a good shape. So all we should comply with all quality management in terms of lab, in terms of manufacturing, in terms of documentation, along with our clinical research. So QA/QC part, this is the principle, and QA/QC is the practice that has to be in the place. I think I'll skip it because this part will be spoken by other speaker. This is the same thing. Now, who should comply with the GCP? Everyone should comply it. Investigators, sponsors, and monitors, and all CROs should comply the GCP. But ultimate responsibility is for the sponsor only. And how do we comply this GCP effectively? For this, we have to ensure we should have the good standard operating procedures. There should be oversight responsibility. There's adequate education and training to the trial subjects, trial investigators, and their supporting staff. Lines of communication should be intact. There should be auditing, monitoring, and there should be discipline to be enforced, and response and prevention should be there. These are the way to make it implemented in the process of clinical trial. The regulatory requirements, these all are, and the regulatory requirements would be different in different setup. There will be local requirements, there will be national requirements, but quality should be maintained by all authorities, and there is a shared responsibility from all. There are codified regulatory requirements like DDA in Nepal, Department of Drug Administration, for investigation of drug only, but not for the whole trial, and like FDA Code of Federal Regulation in US, and EMA for Europe, Food and Drug Administration in China, Minister of Food and Drug Safety, MFDS from Korea, like this. And best practice in industry standards should be ensured by the standard operating procedures. And whatever we do, whatever guideline we follow, these are the guidelines for us only. They will tell how to conduct. They do not tell you how to conduct your clinical trial, but they will define what are the essential requirements. So we have to keep in mind that these guidelines are guidelines for us only, but whatever is required, we have to do it according to the guidelines. And standard operating procedures should be there. If it is not there, then there will be high risk of the misconduct. So there should be misconduct or high risk of non-compliance with the GCP. So SOP should be there in place, and it has to be approved by the IRC as well. The monitoring should be there in between. Otherwise, accurate and complete verifiable data will not be obtained, and human subject will not be protected, and we may not comply with the GCP and regulatory requirements of the trial site. Auditing should be there to make that our trial go on smoothly with the quality data. And the audit types will be different. There could be external audits from regulators and sponsors. There will be internal audits. There will be audit from the CROs or suppliers. Investigator side audits will be there. Regulatory bodies will be doing regulatory audit as well. And if there is anything, any discrepancy or any mishaps, any events, then there could be appropriate external, internal audit for the particular cause only. There could be conflict of interest, and that has to be minimized. And it has to be assured by the GCP or regulatory bodies, and the constraints of the COI will be there on the quality of the trial and the protection of the human rights. And quality audits should be there. There should be third-party or internal audits, regulatory agency inspection should be there, monitoring, and whistleblower will also help us to minimize conflict of interest and will ensure our compliance. And these are my references, and that is all about my presentation. And good clinical practice is, in fact, in our understanding, this is a good clinical research practice with which physicians will be misnomer it like it is our clinical practice. Clinical practice is different and clinical research is different. Thank you very much.
Thank you, Professor Baral, for that very informative overview on good clinical practice. If there's any questions that you need to direct to Professor Baral, please use chat feature. At the end of this discussion, we have few minutes scheduled to answer questions. I will direct them to Dr. Baral for answering. The next speaker on the agenda, or the next speech on the agenda is with Professor Dingle, and it's about confirming to the overall ethical principles in good clinical practice. Over to you, sir. So you need to unmute the microphone,
Yes.
please.
Am I audible now?
Yes, you are, sir. Absolutely.
Are my slides seen? Can you see my slides?
We can see them. It will be better if you can go to the full screen.
Is it fully?
सर तल यो 73% नजिकै छ नि सर एउटा कप जस्तो चिन्ह हजुर यतापट्टि सर यता यता माइनस हजुर त्यो माइनसनेरी हेर्नु त सर हो सर हो त्यसमा हजुर त्यसलाई क्लिक गर्नु हजुर 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 हुन्छ सर अब नेक्स्ट गर्नुभयो हुन्छ सर
आएको छ? Is it full screen now?
हजुर सर
मैले देखेन फेरि
जुमलाई चाहिँ अलिकति घटाउनु पर्छ सर जुम बढी भयो जस्तो
I think there is some problem. Let me fix it. Okay, is it okay now? Is it okay?
You can go with this, sir, but if you go to the lowermost part of your screen on the right-hand side, there's three icons very close to the percentage sign, and the most right-hand sign needs to be depressed there, next to the book and the screen.
Next to the book. But I cannot
very low at the lowest end of your screen.
This video of
on the right hand side, sir, the other side.
हजुर त्यो माइनस अलिकति यो त्यसलाई 58 जति गर्नु न सर माइनस गर्नु न स्क्रिन The 73 छ हैन त्यही
Yeah,
तलै
go
छ
down
सर
a little bit, sir. Go down a little bit. Go down.
No, it's, the lower view is obstructed by the videos, no, of the participant and all those in the lower this thing. So maybe I can.
So first you have to open your presentation separately, then you start sharing, then only you can access to those icons mentioned by Dr. Sang.
I have opened the screen, but
We can actually see that.
सर को 73% छ अलि घटाउनु न सर त्यसलाई 60
Exactly.
70
That's there. That's there.
slide
that's
गरे
there.
ठाउँमा चाहिँ उ आयो के Zoom को.
हजुर हो अलिकति हो घटाउनु त सर त्यसलाई
I think we'll continue like this. We'll continue this mode.
हजुर नभए
That's
यसै
one. That's the one, sir. That's the one.
यो उसको Zoom को तल आयो के मेरो PowerPoint को तल त्यसले गर्दा डिस्टर्ब गरे.
We can continue like this, sir.
Okay. So welcome again, and I'll be talking about this clinical research conforming to overall ethical principles. Now I think it's full screen now. Is it okay?
Yes, yes.
Okay. And then the first core principle of GCP, Professor Balal was talking about is the clinical trials should be conducted in accordance with the ethical principles that have their origin in the Declaration of Helsinki and that are consistent with GCP and the applicable regulatory requirements. Actually, this GCP compliance provides the public assurance that the rights, safety, and well-being of the research subject are protected and respected, and they are consistent with the principles mentioned in the Declaration of Helsinki of World Medical Association and other internationally recognized ethical guidelines, and ensure the integrity of the clinical research and the data. The first core principle is the of the GCP is about the basic ethical principles. I'll be talking about those, and we have twin pillars of protection for the rights and the safety and the welfare of the human subject and participation in a research. That is one by independent review by ethical committees like Professor Baral just talked about. And the other one is the proper informed consent taking process. In a good protocol review, we have science, we have ethics, and we have informed consent. These three components are very important. So we have to review the science. It should be, study should be very well designed and then scientifically sound and robust. And similarly, there should be ethical considerations and then should be following the ethical principles of dignity and respect, beneficence and non-beneficence, privacy, confidentiality, and then finally, there is the issue of proper, informed, understood consent, where we have different components to comply with. In case of informed consent, the key elements are communication, understanding, and ability to ask, and voluntariness, and documentation of the informed consent process. This is the core principle of GCP. The research involving humans should be scientifically sound and conducted in accordance with the basic ethical principles which have their origin in the Declaration of Helsinki. And the basic ethical principles of equal importance are respect for the person, autonomy of the person, beneficence, and we have to judge about the risk-benefit ratio, and the justice. These three permeate all other GCP principles. This principle one is applied in research by design and approval of the protocol by ethics committee, by proper informed consent, where respect and autonomy is respected, scientific and ethical review of favorable risk-benefit assessment. Beneficence should be there, and the fair and transparent procedure and outcome in the selection of research subject. So justice will be complied there, and compliance with national and international laws, regulation, and standards. Basically, the ethical principles are established from the World Medical Association Declaration of Helsinki and the Council for International Organization of Medical Sciences, SIOM's International Ethical Guidelines for Biomedical Research involving human subject, and other guidelines. These are the codes or the guidelines available, like in 1947 Nuremberg Code, and then Helsinki Declaration of the World Medical Association came in 1964, and Belmont Report in the USA. After that, International Committee on Harmonization, GCP, and then other culture-sensitive National Bioethics Advisory Commission of the US, SIOMS, and Nuffield Council of UK. And the latest 7th revision of Helsinki Declaration mentioned that there should be consultation with family members and community leaders. So the Declaration of Helsinki, I will be going in detail about this Declaration of Helsinki, which is a statement of ethical principle for medical research involving human subject, including the research on identifiable human materials and data. This was primarily addressed to physicians, but all other healthcare workers and the scientists can follow this, and it was adopted in 1964 in Helsinki, Finland. So there are some general principle of Declaration of Helsinki. A physician shall act in patient's best interest when providing medical care. The duty of the physician is to promote and safeguard the health and well-being and the right of the patients. Even the best proven intervention must be evaluated continually through research for their safety, effectiveness, efficiency, accessibility, and quality. Medical research should be conducted in a manner that minimizes possible harm to the environment, and appropriate compensation and treatment for subjects who are harmed as a result of participating in the research must be ensured. Similarly, while the primary purpose of medical research is to generate new knowledge, this goal can never take precedence over the rights and the interests of the individual research subjects. It is our duty to protect the life, health, dignity, integrity, right to self-determination, privacy, and confidentiality of personal information of the research subject. Physicians must consider the ethical, legal, and regulatory norms and standards for the research involving human subjects in their own countries, as well as applicable international norms and standards. Research must be conducted only by the individual with appropriate ethics, scientific education, training, and qualification, as already mentioned by our previous speaker. And then risk and the burden should be minimized. Medical research involving human subject may only be conducted if the importance of the objective outweighs the risk and the burden to the research subject. So measures to minimize the risk must be implemented. When the risk outweighs the potential benefit, or when there is conclusive proof of definitive outcomes, the physician must assess whether to continue, modify, or immediately stop the study. Similarly, we have to protect the vulnerable groups and the individuals, and they should receive specifically considered protection. Medical research with a vulnerable group is only justified if the research is responsive to health needs of the priority of this group, and the research cannot be carried out in a non-vulnerable group. In addition, this group should stand to benefit from the knowledge, practice, and intervention that result from the research. Scientific requirements and the research protocols. Medical research must conform generally accepted scientific principle, be based on the scientific literature, and should have adequate laboratory and animal experimentation. However, the welfare of the animals used in the research must be respected. The design and the performance of each research study involving human subject must be clearly described and justified in a research protocol. The protocol should contain the statement of the ethical consideration involved and should indicate how the principle in this declaration have been addressed. The protocol should include information regarding funding, sponsors, institutional affiliation, potential conflict of interest, incentive for the subject, and information regarding provision for the treatment and the compensation in case of harms. Regarding the research ethics committee, Each protocol must be submitted for approval to the appropriate ethics committee before the study begins. And this committee must be independent. It must take into consideration the law and the regulation of the country in which the research is to be performed, as well as applicable international norms and standards. The ethics committee must have the right to monitor the ongoing studies. The researcher must provide information about any serious adverse event. And then if there is any amendment, this amendment has to be through the ethics committee. The researchers must submit a final report to the committee, and precaution must be taken to protect the privacy of the subject and the confidentiality of the personal information of the research subject. Like regarding the proper informed consent, the participation in the research must be voluntary, although it may be appropriate to consult family members and the community leaders. Each subject must be adequately informed of the aims, methods, sources of funding, any possible conflict of interest, institutional affiliation, and the risk and benefit post-study provisions. And the subject has the right to refuse to participate in the study or to withdraw the consent at any time, even after participating in the study. After ensuring that the potential subject has understood the information, should the subjects freely given informed consent, preferably in writing, that has to be signed and dated. The physician must be cautious about the dependent relationship with the physician or may consent under duress. In such situation, the consent must be sought by a qualified individual who is completely independent of this relationship. For a subject who is incapable of giving consent, the physician must seek informed consent from the legally authorized representative, LAR. These individuals must not be included in the research study that has no likelihood of benefit for them unless the research cannot be performed with persons capable of providing informed consent. And the research entails only minimal risk and minimal burden. When a potential research subject who is deemed incapable of giving informed consent is able to give assent to decision about participation in research, the physician must seek that assent in addition to consent of a LAR. The potential subject's dissent should be respected. Research involving human subjects who are physically or mentally incapable of giving consent, for example, unconscious patients, may be done only if the physical or mental condition that prevents giving informed consent is a necessary characteristic of the research group. In such circumstances, the physician must seek informed consent from the legally authorized representative. The refusal of a patient to participate in a study or the patient's decision to withdraw from the study must never adversely affect the patient-physician relationship. For medical research using identifiable human material or data, the physician must seek informed consent for its collection, storage, and/or reuse. Use of placebo, the benefit, risk, burdens, and effectiveness of a new intervention must be tested against those of the best proven intervention, standard of care, except in the following circumstances. Where there is no proven intervention exists, the use of placebo or no intervention is acceptable, or where for compelling and scientifically sound methodological reasons, the use of intervention less effective than the best proven one, the use of placebo or no intervention is necessary to determine the efficacy or the safety of an intervention. And the patients who receive any intervention less effective than the best proven one or placebo or no intervention will not be subject to additional risk of serious or irreversible harm as a result of not receiving the best proven intervention. So we have to have publication and dissemination of the results. In advance of a clinical trial, sponsor, researcher, and government should make provision for post-trial access for all participants who still need an intervention identified as a beneficial in the trial. This information must also be disclosed to participants during the informed consent process. Every research study must be registered in a publicly accessible database before the recruitment in the trial registries. Researchers, sponsors, and publishers all have ethical obligations with regard to the publication and dissemination of the results of the research. Even the negative and unconclusive results should be published, otherwise made publicly available. And sources of funding and conflict of interest must be declared in the publication also. Unproven intervention in clinical practice can only be indicated if there is no effective intervention. So for more detail, we can go to this Nuremberg Code, WMA Declaration of Helsinki, Belmont Report, and other resources that we have. So this is all about ethical consideration. Thank you very much. Thank you. Over to you, Dr. Indu.
Thank you, Dr. Dingal, for that very comprehensive discussion about the topic. The next speaker, the next speaker is Professor Seda Marsuk. She's the chairperson of obstetrics and gynecology at PIMS Islamabad, Pakistan. She's the vice chancellor at Ali Bhutto University at Islamabad and chairperson of Society of Obstetrics and Gynecology of Pakistan. She's also a member of academic board and past chief editor of Annals of PIMS and JSA for medicine. She's going to talk to us today about quality assurance and monitoring of clinical trials and including good clinical practice. Over to you, Professor.
Thank you, Dr. Sankar, for introducing me. I'm not getting the screen share option. Would Dr. Ganesh please share it for me?
Okay, ma'am, I'll try. Is it visible now?
Not yet. Waiting.
Is it seen or not?
No.
No? Okay, I'll
please
again,
use screen share for me. Yeah, please screen share for me.
I'll again. Is it visible now?
Yeah. Yes,
okay.
Thank you. So you'll have to move this for me. I'm sorry.
Yes, ma'am. Yes, ma'am.
So yeah, can we move on, please? So I'll be talking about quality assurance in clinical trials. We all know that clinical trials are collecting data for academia, industry, and for research, and quality management allows sponsors of clinical trials to monitor the progress of clinical trials by the investigators. So quality management is actually assisting us in the efficacy and safety of the understudy disease, illness, or the preventive medicines that we are investigating. And quality management basically ensures that investigators follow the study protocol, they comply with regulatory clinical good practices that we've just heard about, and collect and report quality data. Next, please. So clinical trial quality assurance. So this is basically a CTQA program, and it has to be initiated before the trial so that we set up the systems and processes at the very beginning. Then it has to be carried on during the trial by adequate oversight and monitoring, which can be done both by audits as well as by sponsor inspections. And towards the end, we need to verify that accurate reports are available. So this is a holistic sort of a quality assurance that needs to be administered. Next, please. The goals are basically that we have to have a systematic approach in clinical studies, and we want to ensure subject protection, and we also want to deliver high quality data. Next, please. So basically the objective, the presentation that I'm going to do today is that the clinical research quality is embedded in the clinical trial protocol. So if a robust study protocol is going to be made in which the SOPs are correctly created, we have a good scientific and medical design, we do a pre-assessment and selection of the clinical investigator and site. And if our colleagues who have been involved in clinical research in multi-center research, in research which has been conducted by WHO or by maybe our partners in developed countries, we have a pre-selection and a pre-assessment visit where they come and interview the investigator. They see whether they are capable of doing all the responsibilities that are being planned. We need the regulatory agency's approval and obviously the ethics committee approval. Next, please. So we have to, as our previous speaker has said, that informed consent is a very important part of a clinical trial quality, where the language has to be right. In our country, like, the language is different and the scientific language is different, even regional languages are different. So you have to explain benefits and risks in the right language, which the patient understands. Then again, very important component of clinical trial quality is the investigator meetings and trainings. Like, our department is involved with the London School of Tropical Hygiene and Medicine in doing the Woman 2 trial, and as well as there was a pharmacodynamics trial. So we used to have investigator meetings. There were other investigators in other centers, and nowadays with the availability of Zoom, so we share our experiences, and obviously with the sponsor, we discuss all the problems we have, and the training is basically ensured. Adequate recording and reporting of data is then very important, and unless you have periodic monitoring and audits, you're likely to have major problems in your clinical trial quality assurance. Informed consent, I will not go into the details because it has been dealt with in very robust manner, but basically the correct version of the consent should be used. There should not be any missing consent authorization. There should be procedures which should be performed prior to obtaining consent. Next, please. And again, whenever the inspectors come in to check the clinical trial quality, they assess for whether a financial disclosure has been made, whether the documents are available regarding the protocol, the brochure, the clinical trial agreement, the IRB submissions and approval. Then you have the delegation of authority log. Somebody who's not allowed to do something is doing it without the delegation of authority. Similarly, in trials which are using investigational product, how is it being managed? Is it being documented? The expiry dates, if they are not used, how are they being handled? And the monitoring and auditing reports, they have to be checked whether they are being kept in a proper manner in this particular clinical trial. Next, please. So protocol compliance is again very, very important, because if we are not careful, then the purpose of that study would be lost. The outcome would not reflect the objectives of that study. So any missed visits should be looked into, missed procedures should be looked into. If there's a failure to administer the drug appropriately, or at the wrong time it is being given, in the wrong dose it is being given, then these deviations have to be picked up. Next, please. So we as clinicians and as academicians, we all love mnemonics, and the documentation practices should follow ALCOAC principles. So it's not a very pretty word, but if it is easier to remember, whatever documentation is happening in that clinical research, it should be attributable, it should be legible. Doctors are not very good with legible writing. It should be contemporaneous, that it is written in time rather than retrospectively with remembering. It should be original, it should be accurate, it should be complete. So there are such high requirements to do clinical research that it seems a tall order to follow, but all of us can do it if we take care. Next, please. So whenever you have a visit from the sponsor or from the authority who's auditing you, they check the training manual, they see the site initiation visits, they come and see the various CRF forms that you are completing. Similarly, the processing of the specimens and GCP and protocol amendments are extremely important, and relevant trainings need to be sorted out, and delegation of study tasks to non-study personnel has to be prevented. Next, please. Then there is again very important point that whenever you have a site visit, any unsigned notes, any undated notes, and you're not allowed to use whiteout. It is not permitted. We always cross our entries. If there is a mistake, you let it remain, but you just cross it, but you don't make it so, try to just, so that nobody can read it. The original entry should be readable, but you cross it and move on and change the entry. So these are simple things that need to be looked into, and the subject records have to be evaluated properly so that the clinical trial quality is good. Next, please. The data management is the basic part. The CRF forms have to be timely entry. The data should be per specification of the protocol, and it should be according to the clinical trial agreement. Next, please. So basically, the purpose of the study, as very nicely explained by the previous speaker, is that we have to ensure that the subject is protected and any adverse event is appropriately reported, documented, and annotated by the investigator. Next, please. So whenever there's an investigational product, the lack of patient drug diaries to determine patient adherence or lack of documentation or compliance, all these things have to be very properly documented. Next, please. So we have to make sure that the laboratory inspections and certifications are in there. The specimen, if you have to handle it, it should be adequately handled, and any evidence of inadequate handling has to be reported on and corrected. And the temperature logs for certain drugs which have to be maintained at certain temperatures, how it has been logged, how it has been correctly monitored. These are very important things which have serious effects on your trial results. Next, please. So I'll conclude my presentations by talking about observations requiring actions. This is very, very important. So whenever there's a trial in progress and any deviation or deficiency is observed in the compliance, then you have to make an observation so that anything which has effect on the patient safety, data integrity, it has to be corrected. Supposing you're not doing the right entries, you're missing the training logs, you're not documenting the significant lab results by the investigator taking action. So then what is required? Either it is an action plan or it is a corrective and preventative action plan. Action plan is a very mild activity, while corrective and preventative action plan becomes a little serious. Next, please. So actions can be just completing a delegation of authority log. It was missed and you ensure that it is going to be picked. Similarly, if removing tasks for study personnel that are outside their domain, you ensure that it doesn't happen. And if training has been done but documentation is missing, you can take an action. Next, please. The corrective and preventative action plan, that is the serious problems are happening in the study, and they can pose a significant risk to the rights and safety of subjects and jeopardize data integrity. So a major deviation from compliance to regulations or guidelines or protocol or SOPs requires a corrective and preventative action plan. This is an example can be an incorrect drug dose is being given. There is a lack of investigational product management. The informed consent is not right. You're not informing correctly, and there are major deviations in the protocol. So you have to take a CAPA. Next, please. So to complete your corrective plan, you have to identify the problem, conduct a root cause analysis, identify the problem, develop an action plan, how to correct the problem, implement the plan, and evaluate whether whatever you have corrected, is it working? Next, please. So root cause analysis, I'm sure most of you are aware of this. Whenever you are trying to look into problems, you try to look into the root cause. That can be by brainstorming, by 5 whys, and we'll not go into that details. But we have to look into the cause of the deviations that are happening. Next, please. And once we know that what is happening, then we can correct and implement revisions to documentation. Maybe we need to retrain the study personnel, reconsent the study subjects so that they are appropriately informed, and revise the departmental SOPs, which are leading to these major protocol deviations. And if it is very serious, then we have to inform the sponsor or the regulatory agency that perhaps it is not going as it should. Next, please. So I'll summarize by saying that if you want to do a corrective plan, you have to basically ensure that a good system is set up, there's a friendly compliance review during the study, and you continue supporting the investigators throughout the study. And the best approach is to identify the problem, implement new processes so that the risks are mitigated and they don't happen again. And each event should be basically a teaching tool. Next, please. I would like to thank my resident who helped me with this presentation. And there was, from the internet source, I was assisted by the University of North Carolina, very important common findings report, which helped me to make this presentation. So my final slide is thank you. And I would like to tell you all that in clinical trials, if you wish to walk fast, you walk alone. But if you wish to walk far, walk together. And that's where the success lies. Thank you very much.
Thank you, Professor Masal. That was an excellent presentation, very well thought of and very informative. And I get a lot of messages saying that they learned a lot from that lecture. Thank you so much. The next I'm going to speak to you about the next topic is mine. I'm going to talk to you about including pregnant women in clinical trials. Okay, can you see my slides, please?
Yes.
Yeah, thank you. Okay, so I'll speak briefly about including pregnant women in clinical trials. As the topic today, and as we discussed, a good clinical practice is the international ethical, scientific, and practical standards to which all clinical research is conducted. And the overriding concern there is to ensure that the well-being and the interests of the human subjects who are being studied are adequately safeguarded, as well as you get reliable, correct research data. In principle, the main underlying principles of good clinical practice is beneficence, respect for persons, and justice. So it has to be beneficial, and people should be involved. Any person who is involved should be involved with informed consent, and there should be justice. So it should be the person who is getting involved has to be able to get in or out without being without as an individual, and without restrictions of race, gender, or creed, or origin of origin. Now, when we look at good clinical practice, it's a must adhere protocol, and that's the minimum standard. That's the minimum standard of clinical research that you need to conduct. And we know that the origin of good clinical practice comes way back from the Nazi German trials and the Nazi German clinical experiments that they did in the name of science, and the Nuremberg trials about to correct that. And therefore, because it originates from a very dark place, GCP is very protective in inclusion. And although it's very protective, it's still a minimal standards, and some people have criticized good clinical practice for having a less morally authoritative document than the Declaration of Helsinki, for example. So it has pros and cons of it. And traditionally, pregnant women has been included as a group of vulnerable people because of the fetus that is involved. And hence, there were many concerns about including a pregnant woman and research exposure of a fetus. Because it was the pregnant women were considered vulnerable people or vulnerable population, an inclusion needed a full preclinical animal and non-pregnant women to be studied on the topic before we could include a pregnant woman on the study. And there should have been a benefit to the woman and or the fetus. And if you don't have any benefit individually to the woman or fetus, there could not be absolutely any harm, any foreseeable harm to the fetus. So in other words, the risk to the fetus had to be justified. And obviously you had to use the least possible risk to achieve the objectives, and informed consent from the mother was obtained. And there are certain issues about, okay, if the mother is consenting with about the fetus, what rights or what consenting rights is there for the father comes up. And there are issues about termination of pregnancy research and termination of pregnancy as a result of issues with research. And so generally, the culture has been to exclude pregnant women from most of the clinical trials that went on. Now, why exclude pregnant women? There were multiple reasons to exclude pregnant women. First and foremost, the fear of harm to the fetus and the threat of legal liability. So any harm or any legal liability would be there with the researchers for 18 years or more, and that is something that any researcher would not take quite lightly. And a concern about complicated physiology of pregnant women was used to exclude pregnant women, and uncertainty whether pregnant women will be willing to participate was also a concern. And the regulations which inclusive of GCP, regulations which classified pregnant women as a vulnerable population and a population which needed special protection in research. And sometimes even the institutional research boards and regulatory boards had a little bit of vagueness and ambiguity and restrictive wording on regulations in including pregnant women. And obviously, a research study which include pregnant women is prohibitively expensive to ensure. So the cost was a matter. However, considering all that, the pregnancy itself is now clearly seen as a different physiological state from even being a normal woman. We all know the changes in physiology. We all know that changes due to pregnancy in physiology makes that study subject completely different in pregnancy as compared to of pregnancy. And the fact that at least 60% of women, some people estimate if you take over-the-counter medications, that would be over 90%, will take some medication prescribed or unprescribed for chronic illnesses or conditions that arise during pregnancy. And despite having this situation, extremely few drugs are approved for use during pregnancy. The many drugs that we use are actually never seen clinical trials in pregnant women. And although there are significant physiological changes in pregnancy, including near doubling of maternal blood volume and binding proteins, and pharmacokinetics are not specifically checked for on these drugs for pregnant women. And we all know that animal studies are not really good substitute for predicting results, especially in teratogenicity in human subjects. So majority of research on pregnancy confines itself to issues of the pregnancy and extends to early neurodevelopmental outcomes of the child at the moment. However, although these are things are important, yes, they are important, that there's a new paradigm that is coming in, telling us that pregnancy and pregnancy changes are actually changes that will be carried over for life by both the fetus in epigenetics as well as the mother in terms of her cardiovascular mainly or other system changes. And for example, a woman who is developing preeclampsia will carry that preeclampsia related physiological problems throughout life, including an increased risk of having cardiovascular disease later in her life, as well as that fetus also is known to carry problems throughout its life. So therefore, having pregnant women in clinical research has become an extremely important topic, and not simply because simply pregnancy may unmask chronic disease and may predict future disease and provide opportunity to identify health risks and diseases. So therefore, having pregnant women as a group that to be studied has come into prominence among researchers, and the most compelling reason to justify the inclusion of a pregnant woman in studies is the need for evidence gathered under rigorous scientific conditions that place fewer women with good clinical practice background in a research background that would put few women and their fetuses at risk compared to having these drugs or products that are released without having pregnant women in their studies, who will be exposed after market surveillance, and it will be used by a vast number of pregnant women before the problems are identified if you use aftermarket surveillance as a problem identifier. So that's the most compelling reason to justify inclusion of pregnant women in studies. Therefore, the current thinking from a pregnant population as a vulnerable population, most societies and institutions now think that the pregnancy is actually should be considered as a medically complex population that is to be included in rigorous scientific research with adequate protection from good clinical practice. In summary, poor evidence base for interventions in pregnancy is unfair. So pregnant women should be presumed eligible for participation in clinical studies, and who are eligible should not be excluded just because they're being pregnant. Pregnant women from participation only when there is no prospect of medical benefit to the pregnant woman and the risk of pregnancy and harm to the offspring is known or could be plausibly inferred. And the research interest of pregnant women should be prioritized. Thank you. Thank you. If there's any questions that you can use the chat feature for sending over the questions. The next speaker is Professor Kamath, who is going to talk to us about individual participant data meta-analysis. And he's from Christian Medical College, Vellore, and he's the professor and head of Department of Reproductive Medicine and Surgery. He's also a prolific researcher with extensive research publications under his belt, and an extremely good high index score for his research publications with ResearchGate, and he's also an editorial board member for Cochrane Fertility and Gynecology, associate editor for Human Reproduction Journal, and specialty editor for European Journal of Obstetrics and Gynecology and Reproductive Biology. So over to you, Professor Kamath, for your lecture. We are keen to listen to you.
Okay.
You are still mute, Professor Kamath. I think somebody will need to unmute him.
Okay.
was unmuting.
Yeah. Okay. Thank you very much. I'm audible. Yeah.
Yes, you are. You are absolutely.
Yeah.
Close
Thank you.
Thank you.
Thank you, Professor Asanka and Dr. Ganesh and the whole team here. So I've been patiently listening to the fantastic lectures earlier. So I'll just straight away go to the topic and why it is important that we can talk about this. I'll discuss two aspects, not only IPD meta-analysis, probably also at something called prospective meta-analysis. So I'm just trying to move my slides. Hopefully they move. It's not moving. Yeah. How do you move this now? Yeah. I'm not able to move it. Okay, sorry. Yeah. So I think all the lectures have been on primary research. All of us have talked about randomized control trial, and we know that this is the evidence pyramid, and we know there's something beyond systematic. The top is, of course, systematic review and meta-analysis. But obviously, the primary research is the main contribution for evidence generation. So I'm going to discuss about something beyond systematic reviews, which is slowly coming up, but I think it will be good for us to understand the mechanism behind this. So as we can understand that primary research, as all the talks have clearly elaborated, is quite I think the rules and regulations and all for all of us who are researchers, it's a daunting task actually to put these things together. But if you ask me what's the biggest problem beyond time, I think the funding. Okay, so you require big amount of funding for such kind of big trials with so much of infrastructure, human resources in place. We have the patients, we can do things properly, but there's just huge amount of money required for this kind of research. So what happens essentially is we end up doing our small research in our institution and somehow try to publish it, and the story usually ends there for most of us. So I thought we will go beyond this and how our trials, which are being conducted in academic centers, single trials, if done properly with reasonable care, of course, the ethics and validity of the whole trial still remains, the need for it. How can that be used? So let me just quickly recap the systematic review steps. So all of us know systematic review is the top on the evidence pyramid, and meta-analysis in many cases when we are able to pool similar trials. So these are the steps of systematic review, and I don't want to cap on all these steps, but I would just point out that at some stage, we identify similar trials and try to pool the data from these trials in order to increase the power of our study and get a answer to a research question. Now, these are the steps. So many times we will find a couple of trials, maybe 10 trials, 13 trials, 15 trials, and so on. And we try to pool these trials. Okay, this is called meta-analysis, and that I am sure everybody is aware of. The problem starts when we start pooling trials which are not exactly similar. On the surface, they might look similar, but they are quite different. A trial done in our institution may be quite different from what is done in Delhi or Nepal or Pakistan or Sri Lanka. Even though the PICO, as one of the speaker did mention, may sound similar initially on the surface. So you have to ask yourself this question when you're looking at a systematic review of and a meta-analysis of pooled trials. How similar are the studies, even if they are fitting in for the PICO and the eligibility criteria as met, when we pool these trials? So there lies the problem, and this is called, all the traditional systematic reviews are called aggregate meta-analysis. They will have subtle differences in inclusion and exclusion. You would have described the live birth at 28 weeks. I would have described it at 26 weeks. Somebody else would have done it at 22 weeks, and then you have to pool these trials. Similarly, there could be statistical analysis issues. There will be some heterogeneity in all the trials and quality of reporting. So all this will translate into studies which are looking similar, but we might not be able to get a good valid results after pooling them. And this is again the steps, but I want to just step on the side and move on to IPD at this stage. So what happens in an IPD meta-analysis is we identify the trials, and then we go to the published trial authors and ask for their data. And if they can share their data, that's where it starts. There'll be a very important data sharing agreement and so on. So I'll just go through the steps. So once we have identified those authors who have conducted and published their trials, we can go and ask them for a data sharing. We can request them, and if they agree, we go through something called data sharing agreement, and we try to share an anonymized data set from our trial. Remember that our trial is already published. So what happens is the central team, which is embarked on the IPD meta-analysis, goes ahead and asks the data. And if they ask me, I will go through my institutional ethics board and just ask for permission from my institution. And then we can go ahead and share our data. And then obviously there could be country-specific embargoes, so we need to be cautious about that. However, we can go ahead and share it. Now, what has happened is over some last few years, IPD meta-analysis has come in reality. It's a reality now. But some of the experiences that the authors have, that is these teams have, is many of the times the authors are reluctant to share the data. And there lies the problem. Why? Because some of them don't have the data anymore. It was done quite a years back. Or they are unsure about the validity. There could be legal issues. And then there is more worrying aspect of fraudulent trials being uncovered. So this is the problem I wanted to highlight, because if we do our trials in the right way, even if smaller trials can contribute to the larger science, and that is what IPD can do for us. So once you have the agreement in place, we kind of share our anonymized data. You just put a code for the patients and share some variables. The trial team doesn't need all the variables. They can ask some variables, and we provide that in an Excel sheet, and they will prepare a master data sheet of all the contributory teams across the world. Now, there could be some variables which will be missing, but that is something that can be worked out in imputation. But of course, these central team will do a data integrity check. So if you share your data with them, they're going to run through the basic baseline variable analysis and the main outcomes. And if there are some minor discrepancies, it's fine. But if you have major discrepancies, then there's problem out there. So this is a kind of cross check. Okay, so we have to be careful here that we should be absolutely sure what we are sharing is something that is totally valid. So there are times then the main team will have to have a committee to sit and decide whether the trial is a suspect, and if at all, then we need to find out a way to include or exclude them. Of course, the analysis goes along just like a systematic review, but the main advantage on an IPD meta-analysis is you'll get a data for subgroups. So women less than 35, more than 35, and this kind of data can be extracted. There's something called interaction analysis. I don't want to go into all those details, but those are the advantages. What do the authors who share the data get? They are participating, so they will get an authorship as a team, and that is quite good. Of course, contributing to the greater good of science is the most important aspect here. So this is briefly about IPD meta-analysis, which is going up. As you can see, for 2018, there are 14,000 conventional systematic reviews, and now there are 140 IPD meta-analysis, but they're all picking up. So that's about IPD meta-analysis, but I would also just share a relatively newer concept, which is called prospective meta-analysis, which is something that has come up recently. Now, all that I discussed about systematic review and even IPD meta-analysis is retrospective. That is, the trials have already been done, and now we are going around asking the trial author for data. But prospective meta-analysis works on a slightly different principle. Here we try to, let's say 3 or 4 of us across this continent decide to launch a trial. We can work on a common platform, that is a protocol, and all of us can simultaneously start the trial and have a flexibility to conduct the trial. But we agree to share the data at the end of the trial. And what can happen is you can go ahead and publish your trial, but finally your data also will get published around the same time for the meta-analysis. So what will happen is this will help us have a common protocol. So all the outcomes and everything can be standardized, harmonized. Eligibility criteria can be set in together. And of course, we can work on subgroups as well. So this is a fantastic concept which has come up. And people might ask, why don't we just conduct a multi-center trial instead? But that's not something that is easier to do, because multi-center means that you need larger fundings and all the groups have to come together. The bureaucracy has to come together, and that's not something very easy. So this is somewhere where like individual institutions still can collaborate. And without going to the bureaucracy of joining all together, you can approach your own ethics committee and move on. But all you need to do is harmonize and try to finish the trial around the same timeline. So this is an important paper from Sidler, published in BMJ 2019, where she lays down the steps. And essentially what it does is all of them have started trials together. At some point, they will decide that the people who have finished the trial can publish. And beyond that, the final prospective meta-analysis will come in. So a lot of cancer trials have followed this. So what they have done is in Lancet or JAMA, they have had the main trials getting published in the same issue as the prospective meta-analysis. It's a very beautiful concept which has come up. So finally, this is one of the final slides where I just wanted to highlight this two very important piece of secondary research, which is about collaboration. Reviewers and trials, both team work together. They share common protocol, conduct trials published along with the main trials. This is much more quicker than the IPD meta-analysis. And this is something that all of us can really look at because it will allow us to harmonize and share, because as one of the speakers said, it is about working together. It's very important that we collaborate, because if you do small single trials, they are never going to have that kind of a power to answer a question in a unequivocal manner. That is simply not on, because those trials are very, very large trials, and which is very difficult to put in together. So remember that collaboration is the buzzword here. IPD meta-analysis is very promising. So it basically tells us that let's have a valid trial. Even a good valid small trial in your center can contribute to the larger pool of data, and that's very important. So even if you have come up with small trials, you can still contribute by the IPD meta-analysis if you are ready to share the data. And there is a movement for standardizing definition and outcomes. So this is something in fertility. I know that there is something called CORE initiative, which has taken initiative in trying to standardize what is the agreeable outcome definition. So finally, yes, these two methods are beneficial both for primary and secondary researchers because they provide much more robust results, and it's a win-win for our patients. Thank you. Yeah.
Thank you, Professor Kamat, for that instructive lecture. I think a few minutes has been allocated next for discussion for question and answer session. I'm afraid we haven't got any relevant questions in the chat feature. I can leave the questions, leave that open for questions, but I can't see any questions coming in. Just to fire off one question, Professor Kamat. You talked about the data being not being shared. What do you feel that when is it not good clinical practice to be inclusive at the start, an agreement or to share data if you're conducting a clinical trial? Should we not at the ethics review, or shouldn't it be an ethical review objective to be there?
I would agree, Professor Sanka. The main issue is all these rules and the crystallizing of all these rules have come recently. The concept of sharing data has been quite recent. Whereas when we do systematic reviews or even IPD meta-analysis for that matter, we go back in time. And many of these trials, even in countries where resources were there, were conducted when at a shoestring budget, I would call it. And at that point, like if my PG does postgraduate does the thesis, it's very unlikely that I'm going to keep that data forever, that requires the infrastructure. So that was the reason. But now there is a need to, as you rightly said, at the beginning itself, we kind of agree. And I know that as an editorial member that many of the journals now make you sign the data agreement thing. But it's all upon the journal. Right now, level 2 journals allow only to give a statement, you can share it at a reasonable request. But level 4 journals, it is mandatory that we share our data. So I think it will come in force as the culture evolves. Yeah, on that.
Thank you. Now we are extremely lucky today that we have two very eminent people sharing the platform with us today. Professor Arul Kumaran, who is a world-renowned expert and top person in research, and he has very kindly agreed to be with us today, sir. And Professor Malik Gunavardhani, who is from Sri Lanka, who is an expert on good clinical practice. So both these people, we are very lucky that to have them. And the next few minutes or next part of our discussion or this webinar is about few words of wisdom from Professor Arul Kumaran and Professor Malik Gunavardhani. Over to you, sirs. Professor Malik, Professor Arul Kumaran is there. Can we unmute him, please? Yes. Sorry,
Thank you very much. We were kept muted because in case we interrupt, so that's why we were kept muted. So thank you for unmuting us. Well, it was an excellent seminar, and thank you very much for all the speakers. So all the important points were really discussed, starting from the Helsinki Declaration up to the ethical standards. I think the words beneficence, confidentiality, these are quite important in addition to human rights, because these are the fundamental issues which drive research. So I think there's very little I can add. The only thing I would like to suggest is actually it's always good to consider small groups of trial steering committee, data safety monitoring committee, publication committee, and so on in big research, because then it'll keep everybody under control. Otherwise, people tend to go away a different direction. I just read an article and they said 12 golden rules of GCP. And I'll quickly go through. Know and strictly follow the study protocols. So sometimes that is not done properly. And select and train and keep a log of study team members, record data correctly, ensure adequate study equipment, obtain ethics committee approval, predict recruitment accurately, precisely document product accountability that is came up in the presentations, and check laboratory sample quality, which I think Ganesh's presentation illustrated that, and maintain good trial files and archives. So I think Kamath raised that issue about keeping records, and many of the universities keep records in computer systems, and they make it available for public use, because sometimes you can have the data and analyze it in different ways. So nowadays, raw data is available for the others to analyze, which is also good clinical practice. And keeping everybody fully informed is quite an important issue. So thank you very much. I enjoyed the seminar very much, and all the points were covered. There's nothing very much for me to say other than to say it was a wonderful seminar. Thank you.
Thank you so much for joining us, sir. Professor Malik, are you there, sir?
Yes, so thank you very much for inviting me to participate in this seminar or webinar. So Ganesh, I think you have done a good job by organizing everything, and I certainly don't consider myself a wise man to be giving words of wisdom, but I can just share a few thoughts. I think everything has been covered, and from the absolute basics, which are also very, very important. That is what Dr. Malita covered, was the importance of having a good, solid research question, without which you cannot get objectives, and without which you cannot have an appropriate study design to achieve these objectives. And the conclusions definitely must be on that. Now, that is where the publication comes, which we did not cover today. Everything cannot be covered. Before publishing results, it is absolutely necessary that a proper valid statistical analysis is carried out. Very often what happens is, a lot of the primary research, the statistical analysis is not quite sound, and the conclusions are strictly not so valid. So that is one of the things that we see in published articles. And then we went on with the normal, with the nitty-gritty and the details of the GCP, and wound up with the, I think, very important concept of the IPD meta-analysis and sharing, because the original wording was sharing of, if I remember right, sharing platforms, Dr. Mohan said. So I thought that was very good, because that is the ultimate, because otherwise everyone is doing small bits and pieces here. And that is why today people are encouraged to publish their research protocol of the RCT, so that others can also join. And I think that is the way forward, as you mentioned. So it's that's a very important point that was brought up today regarding the future of these clinical studies. And I think the importance of monitoring also was very well addressed, as well as the overall issues. And especially, yes, Asankar addressed the issue of research on pregnant women. If you don't do research on pregnant women, obviously the pregnant woman will not get the best treatment that she is entitled to. So in which case, we have to do it, of course, with great care and caution. So all in all, I think it was a very good session. And I think Ganesh has to be congratulated on getting all these things together, and all the speakers for doing a great job. And thank you once again for inviting me to make my comments. Thank you.
Thank you, sir. Thank you so much. Next is for the next few minutes is for comments by chairpersons and for the recommendations. Dr. Rohana Hattotua, President of SAFOG. Sir, are you there?
Yes, I am here. Thank you. So actually, I don't have anything to add after the comments by the experts, and so I just wish to congratulate Ganesh and everybody for this wonderful session. As I saw, there were more than 100 participants within the program, so that shows the interest shown by our membership, and even in the last moment the publicity was not enough, but still the message has gone through to our members, and it's very encouraging to see that such large numbers participating. And I saw Professor Hodosi Farhana and Dr. Kusum Thapa from our executive board also participating. So this all gives encouragement. So now we have heard so many learned speakers, listened to learned speakers, and we have gathered so much of information. I think we need to put into practice. So that is what we should do in the term available to us. So we should design one or two research projects so that we will be able to make use of all what we learned, so that it will be very helpful to all of us. So I think the research committee can plan something so that with the guidance and advice of all the other the relevant authorities, so that it could be beneficial to all of us. So thank you very much again to Ganesh, Indu, and all of you for organizing this webinar on research. Thank you.
Thank you, sir. Next, I wish to invite Professor Yusuf Latif Khan to do the vote of thanks and closing remarks. Professor Dinger, can we unmute him, please?
Hi, can you hear me now?
Yes, we can hear you now, sir. Thank you.
So it's been for me a kind of very, very eye-opening seminar, and hats off all the organizers and Dr. Ganesh for doing all this with the help of the SAFOG and the team, and gathering everybody, such a huge gathering, and plus the lectures. We can't thank everybody on behalf of the SAFOG family to be with us on this part of endeavor. And hopefully with all this research, and hopefully we can, unless if you don't publish, you perish. So it's about time that we start writing and getting all our data so that can be published easily all over the world. So I wish all the countries to be very, very gently, and hopefully we can continue such seminars with all your help, and then hopefully we can do that. So on behalf of this, we can advise all of you to kind of thank all the multimedia partners, and for the technical supports which we had a couple of hitches in between, but it was kind of taken care of amicably. So good luck to everybody, and stay safe, and hopefully see you again so soon. Thank you, everyone.
Thank you, everyone, and thank you for the participation, especially our SAFOG officials. Even the FIGO president was here, and our special guests, and all our speakers who were very excellent, and the audience who have been there for more than 2 hours. And I have a small announcement. This Nepal Society of Ops Gyne is going to hold its National Congress in April 1st and 2nd, 2022 in Kathmandu. So you all are invited, and hopefully we can meet in person. Thank you very much. Thank you, everyone, and good night.
Thank you.
Good night.
Thank you.